So this is, the next speaker is Zoé Christenson Wick. She is currently a postdoc at Mount Sinai in the lab of Tristan Schumann. I have known Zoé for many years because she did her PhD at the University of Minnesota, where I am currently. Zoé is studying the role of inhibitory phase locking in spatial memory and epilepsy. She also wanted everyone to know that she makes almost all that she wears. That was apparently her fun fact that was on the spreadsheet I was given. And so the title of her talk is Manipulating Single Unit Theta Phase Locking with PhaSER, an open source tool for real-time phase estimation and manipulation. So I will now pass it off to Zoé.
Okay, awesome. Let's see, let me get my ducks in a row here. Okay, yeah, thank you, Martha, for the introduction. And then also thanks to Open Box Science for, first of all, creating this really cool initiative. I'm an avid supporter of open science, so I think that just making all of it more accessible is really great. And yeah, I'm really excited to share with you kind of the inside scoop on PhaSER, which was recently preprinted. And so yeah, I want to start by just acknowledging and recognizing the whole PhaSER team, in particular Paul Phillipsburg, who is kind of the engineering mastermind behind PhaSER, and Tristan Schumann, my PI, and then all of these awesome scientists, Sophia, Cassidy, Elizabeth, Susie, and Corin, who are the real reason that this project has been able to move forward in the way that it has, to be frank. We have QR codes for the preprint here, and also the code for PhaSER is on GitHub, and I will throw these up as well at the end. Okay, so PhaSER was born of some really interesting data looking at the relationship between the spiking of individual neurons, particularly inhibitory neurons, and oscillations, and particularly theta. So this is just some raw data straight off of a silicon probe that I recorded for this project. And this, I think, just helps sort of demonstrate the complexity, but also the really nice organization of neuronal activity in the hippocampus. So you can see both individual spikes in that sort of dense layer in CA1. And also you can see the theta oscillation really nicely. For people who are not used to looking at this kind of data, it's can be hard to see. So I'll just highlight it here in pink. But yeah, the theta oscillation is this sort of nice, like 6-ish hertz oscillation. It's especially strong there on the border between CA1 and the dentate gyrus. And that's because theta itself largely reflects the excitatory inputs that are arriving into a region, in this case, the hippocampus. And it's well documented that local inhibitory neurons in the hippocampus, and all throughout the brain really, will match their firing to the period when those excitatory inputs that are driving theta are most strong. Here, I think that's shown really nicely. We have a raster of spiking from one putative inhibitory neuron in hippocampus. And you can see that when this neuron is spiking, it's largely spiking around or near the trough of that theta oscillation, which is illustrated on the x-axis. And this theta phase locking is thought to maintain excitatory and inhibitory homeostasis, as well as to organize, coordinate, and facilitate the transfer of information between brain regions within the hippocampus, and all of that is going to help support normal functions of that brain region. So in this case, the hippocampus is largely involved in lots of learning and memory processes, spatial navigation, etc. However, this is all kind of theoretical at this point. The causal role of theta phase locking has been really, really difficult to test experimentally until recently. And the advent of better and faster recording and manipulating methods has really made this start to become possible. And today I'll be showing you how I have been going about exploring the causal contributions of inhibitory theta phase locking in the hippocampus. So yeah, one of the reasons why we think theta phase locking is important at all is because it is altered or diminished in disease models that have well-documented altered excitatory inhibitory balance and or cognitive deficit. And one of those diseases is temporal lobe epilepsy, and that's the context in which I study it. So one of the major questions that I've been trying to ask throughout my postdoc thus far is, does theta phase locking of inhibitory neurons contribute causally to the phenotypes of diseases such as epilepsy? So to answer this question, I use a mouse model of temporal lobe epilepsy called the pilocarpine model. This model really nicely recapitulates the disease as it is seen in humans. And in this model, mice are treated with pilocarpine, which is a muscarinic receptor agonist, and are held in a state called status epilepticus, which is a state of constant or continuous seizing for about 2 hours. They're brought out of that state with diazepam and then are returned to their home cage. And then over the course of several weeks, spontaneously occurring seizures will start to occur, and as well as cognitive deficits. Then in these epileptic mice, as well as in control mice, we can perform head-fixed, acute, in vivo electrophysiological recordings using silicon probes. And that will allow us to record the hippocampal oscillatory activity or the local field potential across all the layers of the dorsal hippocampus, as well as the single unit activity simultaneously in these animals while they are running down this virtual track and earning water rewards. And in Tristan Schumann, my PI's postdoc work, using this data collection method, he found that the theta phase locking of inhibitory neurons in the dentate gyrus specifically of epileptic mice was severely altered. So as you can see in the control mice, the inhibitory neurons' mean preferred firing phase is really nicely clustered around the trough of that CA1 theta oscillation. While in epileptic mice, their inhibitory neurons have preferred firing phases that are more widely distributed across all the phases of this theta oscillation. So this is really quite interesting. And the fact that it's specifically this inhibitory theta phase locking is going wrong in the dentate gyrus is specifically interesting as well. And that's because the dentate gyrus of the hippocampus is thought to act as a sort of gate that filters out a lot of the excitation that arrives into the hippocampus. And typically that excitation is arriving in rhythmic fashion. And like I mentioned earlier, this is one of the main drivers of hippocampal theta. So in control conditions, that inhibition in the dentate gyrus is rising and falling in time with that rhythmic excitation. But in epilepsy, the inhibition is much more dispersed. And we think that this may allow for seizure vulnerability points where the excitatory inputs are still very strong, but are no longer being matched by local inhibition. And so my idea with this work is perhaps that by increasing inhibition in a rhythmic manner so that it realigns with that rhythmic excitation, we might be able to reduce opportunities for pathological seizure activity to emerge. But how do we actually test that? What I need to do is create a tool that would allow me to alter inhibitory neuron spike timing relative to this theta oscillation in real time and measure the impact on seizure activity. So in control mice, what we wanted was to try and shift the inhibition so that it was now occurring at a wrong phase of theta, like the peak. And in epileptic mice, we wanted to realign the inhibitory spiking to the typically preferred phase, which would be the trough, and then measure seizure activity as an output. So we devised a system that would allow us to do just that. We're calling it PhaSER because it allows us to deliver phase-locked stimulation to endogenous rhythms. And the basic premise here is that we can record from the hippocampus in an awake behaving animal, take that hippocampal LFP, and then filter it for theta in real time. When you apply filters in real time, you often run into distortions at the edges of the sampling window. So then we use an established autoregressor to predict about 1 cycle into the future. And then that will allow us to then accurately detect the theta phase using a Hilbert transform. We can then use that phase detection to trigger a light to be delivered at our target phase, whatever that is. And then the light delivery will activate an opsin. In this case, it's channel rhodopsin. The excitatory opsin is typically activated by blue light in somatostatin or parvalbumin expressing inhibitory neurons in the dorsal hippocampus. And what's really key here is that we need to do all of this with very low latency, since what we'd like to do is manipulate the spike timing relative to the ongoing real-time endogenous oscillation in a relatively precise manner. So for those interested, the round trip latency of our system right now is about 3 milliseconds. And for reference, a theta cycle is around 120 milliseconds. So here is a video from our GUI, the PhaSER GUI, which you can download on off of GitHub if you'd like. But to use this, the first thing I do is I just collect some baseline data. So this baseline period is going to just train the autoregressive model. And to do this, I just go in and I hit that acquire baseline button on the right and then let it run for a bit. There's a timer in the status box that's in the upper right corner. And that will indicate how long the baseline has been collected for. For recordings, I typically collect about 2 minutes of baseline, but in the example video I'm about to show you coming up, I'm just using the baseline that was 15 seconds, the one that just ran, and the phase detection accuracy is still really, really stellar. So as that baseline data is being acquired, it's being used to train an autoregressive model, which allows us to, as I mentioned before, predict about 1 cycle into the future when applying our manipulations. And what this looks like is it takes in a 1 second sampling window of LFP data, which has been downsampled to 1000 Hertz, and then applies a 0 phase filter. Using a 0 phase filter here is really important because it will cancel out any phase shifts that would otherwise be introduced during the filtration process. However, this process still does introduce distortions to the signal at the edges. And so we trim those off and then use the autoregression model published in this Chen et al. Paper to forward predict the oscillation based off of that baseline data that we just collected. And then a Hilbert transform is applied to detect the phase of oscillation. And once whatever target phase was previously set is detected, a TTL pulse is sent to trigger light delivery. So this video is going to happen kind of fast. And so let me just get it started and then I'll walk you through it. Okay. So what you will see here is that raw LFP data coming in blue. And this is real time, straight from the mouse's brain, pretty much. And the predicted signal from that's using the autoregression algorithm is in pink. And then the light trigger is in red, those red lines that are going up and down. And here I set the light trigger to occur at a certain phase over here on the left. And that phase is listed in degrees. So if I wanted to target the peak of theta, I could either put in 0 degrees or 360 degrees. And here I've put in 170 degrees, which is just going to target right before the trough. And this video is showing everything in real time. And so you can see there's like really nice, really gorgeous targeting of that red light trigger to the trough of the theta oscillation. And not only that, but also the predicted signal is really nicely matching that raw LFP. And I just want to emphasize again, because of the way that we design the system, it can trigger light delivery to any phase of theta, not just the minima or maxima. You just type in whatever phase you want to target in degrees and it will just send triggers at or around that phase. It also is just sending a TTL pulse. So if you want to set this up with an electrical stimulation or something else like that, you can. Hello, sorry. Oh no, I think my PowerPoint just froze. I'm gonna stop sharing my screen for a moment and then restart. Sorry about that. PowerPoint is not responding, of course. All right, it's back. Thank you for your patience. Share screen. Okay, does that look good?
Yes.
Okay, thank you. Okay, so we have that accuracy and precision data. It's a little bit easier to see on the whole than those videos. So we have that raw hippocampal activity collected from our silicon probe, and then below that we have the theta oscillation, which has been filtered offline in this case. And then in blue you can see our light delivery triggers, which are being targeted to the trough. And below that, in this circular histogram, I have all the light deliveries targeted to the trough, and this is from one animal. So all of the light deliveries that are targeted to the trough in this animal, and you can see that the distribution of those are occurring really nicely at that theta trough, and very few to none are happening at like the opposite phase, for instance, and they're all really targeted around that area. Similarly, it looks just as good when we want to target the theta peak, for instance. But again, of course, these are just examples from individual animals. The group data is shown here. And you can see this also looks really nice. On average, we have our light deliveries occurring around our target phase with an error of approximately 15 degrees. So that's really nice. Now, you may be wondering how the accuracy of the system may be impacted by the oscillation itself, for instance, by the power of the oscillation. So I compared how the theta power in the reference channel that I'm using may correlate with the precision of light targeting. And precision here is shown on the y-axis, and theta power is shown on the x-axis. And you can see that we don't really see a correlation between theta power and precision of this of our PhaSER for targeting either the trough or the peak. But I do want to emphasize here that as theta power would approach 0, we would absolutely see terrible accuracy emerge, because the Hilbert transform is going to work to try and find phases in an oscillation, even if that oscillation isn't actually there. But within these physiological ranges, the light deliveries that are occurring equally precisely during running for both trough and peak targeted stimulation. So now that I think I'm happy, at least, with the accuracy of our light delivery, the next question is, can we actually shift the phase preference of opsin expressing inhibitory neurons? So I started with a subpopulation of inhibitory neurons called somatostatin expressing, or SOM neurons, which will be expressing the excitatory opsin channel opsin. And these inhibitory neurons are highly prevalent in the dentate gyrus hilar layer, and provide strong inhibition to the dendritic fields of dentate granule cells, which are the principal excitatory neurons in the region. I can then isolate those clusters using kilosort and phi, and then find cells that are opsin expressing by delivering pulses of blue light. And you can see an opsin expressing cell example on the right. So then I would follow that cell's activity back to the baseline period that I collect and determine its native theta phase preference. So in these optotagged somatostatin expressing neurons, they show really nice clustering for phase preferences around or near the trough of theta. In the bar graph on the right, you can see how strongly coupled their firing is to one particular phase, which is reported as an R value. And when I applied blue light stimulation to the trough of theta, I saw their phase preference sort of tighten around the trough of the oscillation, and I saw the strength of the phase preference increase. And then, this was the really exciting moment for us, when I applied light stimulation to the peak of the theta oscillation, I saw the preferred phase of these somatostatin opsin positive neurons shift towards the peak. So that was great. It shows that we can actually shift the phase preference of these inhibitory neurons with this phase-specific manipulation. But we wanted to test to see if there were impacts to the LFP, in particular to theta, from doing these phase-specific manipulations to these cells. So we first looked at theta power in the reference channel and saw no significant impacts of the manipulations on theta power. I then looked across a more widespread frequency range and across all the layers of the dorsal hippocampus, CA1, and dentate gyrus that were recorded. And those are shown in these power spectral density graphs. And again, I saw no significant changes in power between baseline trough and peak targeted stimulation. Finally, I wanted to see if these manipulations might be shifting the theta phase, as this is something that I know other inhibitory neurons are capable of doing. And also, if we were shifting the theta phase, it would drastically impact our ability to detect theta in real time. So to test this, I aligned the reference layer LFP to 110 millisecond light pulses and then averaged the traces together and measured the mean LFP envelope in the period before light onset to the period after light onset. This is from an example animal on the left with individual LFP traces in gray, and then I've just highlighted a couple in blue so that they kind of pop out a bit, and then the mean trace in white. And so if the stimulation of these neurons was producing a theta phase reset, you would expect to see this white line start having a strong theta band oscillation to it that emerges after the light onset. And you would expect the mean LFP envelope to increase significantly after light onset compared to before. We don't see either of those things, so this suggests that we're not causing a theta phase reset with our stimulation. So then we wanted to take a look at parvalbumin expressing cells. These are also inhibitory neurons that have, they're also inhibitory neurons. So while somatostatin cells typically inhibit the dendritic arbors of their postsynaptic targets, parvalbumin expressing cells will typically apply their inhibition directly to the somata or axon initial segment of their postsynaptic targets. Additionally, PV cells are capable of firing at much higher firing frequencies. This is going to become relevant later on. And this makes them quite distinct and overall a very powerful provider of inhibition in the hippocampus. So we were curious if our phase-specific manipulation could alter their firing phase as well. And when we did the same manipulations in animals that had opsin expressed in these PV cells, we saw the trough stimulation didn't really change, but perhaps just tightened that or got the phase preference of these cells a little closer to the true trough. But when we did peak targeted stimulation, we saw that it was much less effective at actually changing the mean firing phase of these neurons as compared to the somatostatin expressing cells. So in order to look at what was really going on here, I started pulling out the firing profiles of individual cells. So here's an example. So we can see that at baseline, the firing rate of this parvalbumin expressing neuron increases nicely at the trough as expected, and then decreases towards the peak. And when I stimulate at the trough, I see a nice increase in firing at the trough. Again, this is as expected. But when I stimulated at the peak of theta, while I did see an increase in firing at the peak relative to baseline, I was not seeing a decrease in firing at the originally preferred phase, which would be the trough. So really, this was not quite doing what I wanted it to be doing in these high firing rate cells. What we wanted was to be able to shift our phase preference, not just add more spikes at a different time. And so we figured that a bidirectional optogenetic approach would better suit our needs. So I decided that to try out this relatively new opsin called bipoles, which is described in this paper, this Virak et al. Paper. And this opsin has several cool features. Most important for us, it would allow for manipulation of spiking in both directions relatively quickly and effectively. In this opsin, excitation occurs upon delivery of red light, and inhibition occurs through the chloride channel opsin GTACR2, which is activated by blue light. So I tried this out in vivo, and it worked really beautifully. You can see, here's an example, optotagged PV cell. You can see red light activates this opsin, blue light inhibits its spiking. And importantly for us, this inhibitory stimulus doesn't produce a strong rebound after the light, which is also kind of interesting. So then the idea is that I will apply red light for brief activation at my target phase, so either the trough or the peak, and then inhibit with blue light elsewhere in the theta cycle. And this should, in theory, allow me to control the phase locking of these neurons a bit better. So does this bidirectional manipulation actually change that in these fast firing rate PV cells? So here again, we have baseline in blue. If I activate at the trough and inhibit at the peak, I can exaggerate that phase preference as expected. And so what about when I activate at the peak and inhibit at the originally preferred phase? I can actually shift the phase preference of this neuron. I increase it at the peak and decrease it relative to baseline at the trough. So this is working well now. So now we can finally test our hypothesis, which is to remind you, was that misaligned inhibitory spiking was contributing to seizure activity in epileptic animals. And so my experiment is to compare if changing the phase preference of these inhibitory neurons in otherwise healthy animals could exacerbate seizure activity by moving their firing to the peak of theta, or in epileptic animals by moving their firing, realigning it to the trough, could I reduce seizure activity? And so one of the ways that we've tested that is using an acute seizure model. So I took both control and chronically epileptic animals and performed an acute head fixed recording and collected some baseline period, and then injected them with systemic chymonic acid, which will induce a seizure. So immediately after the chymonic acid injection, I begin my bidirectional closed loop stimulation, either stimulating at the trough or at the peak of theta. And then I measure the latency to the emergence of that first seizure. And in otherwise healthy mice whose inhibitory phase locking is intact, I don't see any difference in seizure latency between the trough targeted stimulation and the opsin negative animals, which is as expected. But I did see a significant reduction in the time to that first seizure when I misaligned inhibitory neuron firing to the peak of theta. And the epileptic data is much more variable, which I think is kind of interesting. And we're collecting more to flesh this out a bit. So this is preliminary. And it's hard to say anything too strongly about this, but I think certainly in the control animals, that misaligning the inhibitory spiking to the peak is decreasing the time to that first seizure by an average of about 5 minutes, and is therefore increasing seizure susceptibility in otherwise control animals. So I think I'll wrap up there. So I've walked you through PhaSER, the closed loop system I developed with Paul Phillipsburg, a grad student in the lab. And this allows us to deliver manipulations in real time to ongoing endogenous oscillations. I want to emphasize that PhaSER is really flexible. You don't need silicon probes to record your oscillations. You can use whatever recording method you would like. And you also don't need to use light to apply your manipulations. You could use an electrical stimulation or anything else that you can trigger with a TTL pulse. So if you're interested in applying this, you can find all the info on the preprint or on GitHub, or reach out to me, Paul, or Tristan. I've also shown that these bidirectional manipulations are really helpful in altering the phase locking of inhibitory neurons, especially those that have a higher or the metabolic ability to fire at faster basal frequencies. And this is a good moment to mention that while the preprint doesn't actually mention those bidirectional manipulations, the PhaSER code that's on GitHub is fully up to date with options for those manipulations. And finally, I shared some promising preliminary data suggesting that theta phase locking may impact the emergence of seizures. So with that, thank you for your attention. And thanks to my lab mates, especially Paul, for all the hard work on this. My PI Tristan Schumann and our collaborator Denise Kai for invaluable guidance on this project. And thanks to our funders for keeping the lab and this project afloat. Yeah, thank you.
Great, thanks Zoe for a really cool talk. So we will open it up for any questions. So you can either just go ahead and unmute, you can raise your hand, you can type it into the chat. Anybody have an initial question? Because I have a question. So I'm wondering about in just the kind of standard phase locking, not necessarily the manipulation, is there anything to suggest a progression of that misalignment that might, or a difference in that misalignment depending on the behavioral state. I'm thinking about data about both just that theta can change based on if the mouse is running or at rest, and then also seizure susceptibility and locomotion states in terms of how often the mice are moving versus when they're seizing. So like, is there any heterogeneity in that misalignment depending on either the behavior or whether the mouse is having a seizure or approaching a seizure or something like that?
Yeah, so in that initial, in Tristan's initial paper where he described the loss of phase locking in these and the inhibitory neurons in the dentate gyrus, he also found that this was maintained whether or not the mice were running. So theta does become much stronger when animals are running versus when they're not. But this sort of misalignment was maintained. And so it didn't seem to be super state specific in that way, although I'm not sure about like, those were all like awake animals that were, in virtual reality, across different states like sleeping. And I don't know the answer to that yet. I didn't get to mention this, but one thing that we built into PhaSER that I think is kind of a cool, it has a cool relationship to your question, is that we have built in this sort of toggle switch, which allows you to choose whether or not you're going to restrict your manipulation to when the mouse is running or when it's not running. And so for us, that's like, whether or not the mouse is running on this ball in virtual reality, but this could be adapted to any input that you would want to threshold to like turn your manipulation on or off based on the mouse's state. So that could be movement or, use your imagination, it could be really anything. So, yeah.
Very cool. Other questions?
Hi, Zoe. My name is Felipe. I don't actually have a question right now. I just would like to express that was a very excellent talk, and I am very interested in implementing PhaSER. So I will reach out to you later, and we can keep the conversation going. So thank you.
Fantastic. Please do.
I think we have a question in the chat.
It might have been sent directly to you. I'm not seeing. Oh no, sorry. It must have been earlier in the talk. There was a question that said, are your graphs from data taken only when the animal was in status? So I guess the initial phase locking, potentially that information.
Yeah, so none of the data shown is from an animal while it's seizing or in status. So all of that phase locking data is from is basically the interictal periods between seizures. Yeah. Oh, and Martha, the second part of your question that I ignored earlier and then just was reminded of, I do have some like very preliminary data to suggest that how recently a seizure happened will influence how off the phase locking of these inhibitory neurons is. I need to flesh that out a bit more, but I do think that there seems to be a relationship between the time since a seizure that would have just happened in an epileptic mouse on its own and how badly phase locked the inhibitory neurons are.
Oh yeah, that would be really interesting. Yeah, because that's what I was thinking about, is like the immediate, immediately pre-ictal period versus then the immediate post-ictal period, like what kind of those relationships would look like or things like that. Yeah. All right, so if there are no other questions and we're approaching time, I guess I'll say thanks, Zoe, and then of course thanks to Daria as well for really interesting and cool epilepsy and excitability based talks today. And thanks so much to Open Box Science for the invitation for me to moderate this session. This was really a really cool experience. So and thanks to everyone for attending and for all your attention.